Biotechnology Principles and Processes Part 1. In this video, you are going to study about recombinant DNA technology. Biotechnology deals with the techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. Old biotechnology refers to activities where the types of products obtained have remained the same as those obtained from the natural strains or cell lines. The production technologies based on genetic engineering are called modern biotechnology. Therefore, keeping in view both old biotechnology and modern biotechnology, European Federation of Biotechnology has defined biotechnology as the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. The core techniques that enable the birth of modern biotechnology are genetic engineering and maintenance of sterile ambience. Now, genetic engineering refers to techniques to alter the chemistry of the genetic material, that is DNA and RNA, to introduce these into host organisms and thus change the phenotype of the host organisms. Maintenance of microbial contamination free or sterile ambience and chemical engineering processes to enable growth of only the desired microbe or eukaryotic cell in large quantities for the manufacture of biological products like antibiotics, vaccines, enzymes, etc. Gene cloning is the process of synthesizing multiple copies of a particular DNA sequence using a bacterial cell or another organism as a host. The gene of interest is inserted into a self-replicating DNA molecule and the resulting recombinant DNA molecule is amplified in an appropriate host cell. Now let us look at recombinant DNA. So recombinant DNA is DNA from two different sources that have been combined in vitro. There are three main reasons for creating a recombinant DNA, namely to create a protein product, to create multiple copies of genes, and to insert foreign genes into other organisms to give those organisms a new trait. Recombinant DNA technique permits the isolation of desired gene from any organism and its transfer and expression into the organism of choice. Genetic engineering is a popular term for recombinant DNA technology. Now let us look at the applications of recombinant DNA technology. This technique is used to produce transgenic organisms that possess novel capabilities, that is to obtain novel pharmaceutical proteins. For instance, human insulin is being commercially produced from a transgenic Escherichia coli strain that contains and expresses the human insulin gene. Through recombinant DNA technology, a large quantity of recombinant proteins can be produced. These proteins are produced by the specific transgenes that were introduced into them. A number of these proteins of great medicinal value are now produced on a commercial scale. Let us look into the steps of gene modification. In 1972, Stanley Cohen and Herbert Boyer invented the technique of DNA cloning, which allowed genes to be transplanted between different biological species. They isolated the antibiotic resistance gene from Salmonella typhimurium plasmid. When they transferred this gene into Escherichia coli, the latter exhibited antibiotic resistance. The cloned genes were not only stable in E. coli, but the genes were expressing. Hence, DNA molecules from foreign organisms had been transcribed and translated in E. coli. The cutting of DNA at specific locations became possible with the discovery of molecular scissors, the restriction enzymes. A plasmid was used as a vector to deliver the alien species of DNA from Salmonella typhimurium to the host organism, that is Escherichia coli. The linking of antibiotic resistance gene with the chromosome of E. coli was done with the aid of DNA ligase. When integrated in this way, the antibiotic resistance gene replicated along with the E. coli chromosome using the DNA polymerase of the host, that is E. coli. and this technology was a major breakthrough for genetic engineering. Hence, we can summarize the steps of genetic modification as: first step would be identification of DNA with desirable genes, then introduction of the identified DNA into the host, and lastly it is the maintenance of introduced DNA in the host and transfer of DNA to its progeny.